when new tools are used together and provide congruent results, we have increased confidence in our ability to detect important populations for management consideration. I applied phylogeography to Hydromantes shastae. It's one of 3 species of Hydromantes located in North America. The Shasta Salamander is a terrestrial web-footed plethodontid salamander. It primarily has a hotspot distribution in that in certain localities it's very easy to find in high abundance, but in other localities it's seemingly non-existent. The large populations are primarily associated with limestone outcrops, this being a larger one. Also, small limestone outcrops can support large populations as well. The IUCN lists this species as vulnerable, and the state lists it as threatened, and that's primarily due to its highly restricted geographic range, which is located primarily just around Shasta Lake, which is just north of Redding in California. And the lake itself is a reservoir. The dam was built in the 1940s. During this time, it flooded 4 major river valleys. This likely impacted many of the pre-existing populations. The reservoir itself is an important source of hydroelectricity and also recreation. Plans to increase the revenue from these 2 sources are in effect, and some of them actually estimate a potential raise in the dam of up to 200 feet. Certainly raising the lake level of water up 200 feet would inundate a lot of the populations that are known, likely submerging them, but also reduce the available habitat for this species dramatically. Another important practice in the area is limestone quarrying and mining, which directly degrades the available habitat for these outcrop-related species or related individuals. So with these threats in place, there's a strong need to assess the genetic differentiation of this species and to evaluate the distinctiveness of many of the populations located throughout its range. And I started to do this by analyzing mitochondrial markers. So mitochondrial markers, they're on a more recent time scale than nuclear markers due to their smaller effective population size. And I looked at 2 genes. I sequenced roughly 788 base pairs of the cytochrome B gene and 530 base pairs of the 16S gene, and I analyzed them in a Bayesian phylogenetic analysis. And in this analysis, if populations are separated for long periods of time, then the analysis should show high levels of phylogenetic support throughout the tree and also high levels of genetic differentiation between the different clades. And this is exactly what we see. On the left, we have the tree, which shows what highly supported nodes as squares, and it shows support for 5 major branches of Hydromantes. And it also shows high support for many of the subclades between them, indicating differentiation. And when we look at this on the map, we see that there are 3 widespread clades of this species of salamander, and 2 clades that are identified just as single populations in red and blue, showing them to be much more divergent than the rest. And what I'd also like you to get from this slide is that within these clades, differentiation is low. But between clades, genetic differentiation is actually very high, remarkably high, in fact. Yet the habitat between these is primarily continuous and seemingly suitable. So this level of differentiation is remarkable and a little bit difficult to understand. And we can see this highlighted in this figure. So for the central pairwise comparison between the blue and red population, we have roughly 5% genetic divergence in cytochrome B over roughly 2.5 miles. This is staggering. It's huge. And in the other pairwise comparison, we have, again, over 4.5% divergence over just 2 miles. And again, this is continuous habitat, so this is incredible divergence at really small spatial scales. And when we look closer at Potter Creek, the red circle, we see that it's roughly 4.5 to 5% divergent from every other population in this study. So it alone, a population right in the middle of the range, is totally different from all other populations of Hydromantes shastae. But looking at just mitochondrial data isn't enough. Now with new molecular markers, we can analyze other data sets as well. And so I analyzed a previously collected allozyme data set from Dave Wake and others that was collected in the 1970s and analyzed it with some old and some new methods. Allozyme markers are nuclear markers, so they're on a little bit different time scale than mitochondrial data sets. And each allozyme can reflect a different temporal scale. And what we're kind of doing is looking for patterns in those to reach some kind of congruent conclusion. 18 allozymes were collected then, and we'll be in this analysis. And what we're looking for in this analysis of Alzheimer's is that if populations have been separated for long periods of time, they should show unique or fixed allelic differences to other populations, and certainly different proportions of these of shared alleles. And again, this is exactly what we see. So for this first Alzheimer's, what we'll do is just look at the pattern that it shows. And what we can see is that there's two alleles. There's 5 populations fixed for the black allele, 3 populations fixed for the green allele, and Potter Creek, that population in the middle, falls intermediate between the two. For this second Alzheimer's, we can see that there's primarily a green allele distributed throughout the species, but that the two populations in the center, Campbell and Potter Creek, share a strong link with this blue allele. And in this one, again, we see a shared link between the two central populations with the red allele. But this next one, this fourth slide, shows probably the most striking example of evolutionary divergence with these Alzheimer's, in that it shows Potter Creek having a unique fixed allele, the red one, compared to all other populations which are fixed for the black allele. So this indicates an incredible amount of isolation and divergence within this nuclear data set. And we could do this for all 18 of the loci, but that would take a lot longer than I have. So one nice way to kind of summarize this data is through a summary statistic like Ney's genetic distance, D. And in doing that, looking at pairwise comparisons from Potter Creek, we can see that pairwise comparisons of Ney's genetic D have values of roughly 0.2 or higher to at least 6 of the 8 comparisons we can make. Now, historically, a value of 0.15 has been used by certain researchers as a species level cutoff. Now, if you're going to take a little bit more conservative approach and not designate it as a species, 0.2 is certainly a very interesting value. It certainly indicates a lot of isolation, a lot of divergence, so that really Potter Creek stands out as an important population. Another way that we can analyze these two data sets is through multidimensional scaling. And multidimensional scaling is a way to assess the pattern of relatedness across the landscape. And it clusters related populations based on pairwise genetic and geographic distance matrices. And when we look at both of these data sets with multidimensional scaling, what we're hoping to see or trying to find is basically similar clusters. So if it's grouping the same populations together, then those are really going to be the distinct historical entities and ones that would be important for management. So we can see here that the Alzheimer's data set picks out 4 major groupings of populations, 2 of which are identified just as single populations. And we can see with the mitochondrial data set that it picks up 3 major groupings. And when we put the 2 data sets together, we can try and synthesize what they're telling us. And what we can see is that they both highlight a large, widespread orange clade, a blue clade. They both pick out Potter Creek, the red population, as distinct. And the Alzheimer's data set also picks out the green population. Unfortunately, we don't have that population in the mitochondrial data set yet, so I can't say anything about that at this time. And a final way that we can analyze some of the data sets that we have for the species is through a Bayesian analysis of population structure. And the program BAPS defines the optimal number of groupings for a certain data set. So basically what it does is takes all the individuals and color codes them based on who else they're related to. And the nice thing is that we can see which populations they came from, but also see which other ones that they're related to. And while the program is designed to define an optimal number of groupings, what we can do is actually hierarchically test different levels of relatedness by forcing different numbers of groupings. And what we're trying to see in this test is that if populations are identified as discrete entities, then they're again going to be the distinct evolutionary units important for management, and that groups of populations are important for management as well. So what we can see here in this first slide is that if we're going to look at all the individuals within Hydromantes shastae, they're all going to be more related to each other than they are to a different species. So a grouping of 1 would group all of Hydromantes shastae together. Now, if we were to look within that and see which individuals more grouped together, we see that 3 populations contain individuals that are more grouped to each other than they are to the rest of the blue populations. when we split it into 3 groups, we see that now it's starting to pick out some of the clades that we saw in the mitochondrial data set as well, so that we have the green kind of in the middle of the range as a distinct clade. We have a yellow and a blue. And what happens next is probably the most interesting, and that is when we force it into 4 groupings, we see that Potter Creek, the red square, again pops out as this unique entity, as a distinct unit. And we can see that in doing that, it also picks out the green square above that as a unique population. And that's the most interesting thing about this, is that these are populations. These are single sites that are so different from the rest that they're treated as their own entities. When we look at 5 groupings, we see that on the east side, it picks out another population. And technically, the program defines 7 population or 7 groupings as the most important, but as the optimal number of groupings. But realistically, what's forcing 6 and 7 doing is just picking out divergent individuals within the same population. So really, k equal 5 is the most important number that we're looking at. And the most important take-home message from this is that it is picking out these divergent clades and divergent populations, and again, re-corroborating a lot of the data and results that we had from previous analyses. And the management implications from this are that we have striking evolutionary divergence over remarkably small spatial scales. Throughout the range of Hydromantes shastae, we have divergent populations and lineages with extraordinary differentiation. And there are threats. Rising lake levels and mining practices are a serious concern for many of these sites. So Hydromantes shastae deserve strong management consideration if we hope to conserve evolutionary history and evolutionary potential in this unique species. For this research, I'd like to thank a bunch of people, particularly my advisors, Dave Wake and Craig Moritz, the Forest Service for funding, and a bunch of other people that were influential in this. Thanks for your time.